Fermentation, Cellular Respiration by Zohar Hassan and Milagros Santos. So, as discussed previously, in the previous presentation, we were talking about cellular respiration under aerobic conditions. And as mentioned before, the first step in cellular respiration is glycolysis. And glycolysis is an anaerobic process, so it can either happen in aerobic conditions or anaerobic conditions. So, because none of its 9 steps involve the use of oxygen. However, immediately after glycolysis, the cell must continue respiration in either condition, either aerobic or anaerobic. And this usually depends on the circumstance of the cell. A cell that can perform aerobic respiration, which means it has the presence of oxygen, from glycolysis, it will continue on to the citric acid cycle in the mitochondria. However, if it is unable to perform aerobic respiration, then it's in a situation where there is no oxygen, then it will move into a type of anaerobic respiration. So either lactic acid fermentation or alcoholic fermentation. These fermentation, basically what that is, for example, some cells like yeast, they're unable to carry out aerobic respiration, so they will automatically move into a type of anaerobic respiration called alcoholic fermentation. And fermentation is the process of cellular respiration to produce energy without the presence of oxygen. So, in our experiment, we pretty much focused on yeast and how it undergoes fermentation without the use of alcohol, without the use of oxygen. So basically, in both aerobic and anaerobic respiration, NADH is part of the enzyme complex and has to be restored to NAD. It has to be oxidized, which is its oxidized state. If there are aerobic conditions, the NADH molecule can be transported to the mitochondria where it can be converted back to NAD, and this plays a role in the electron transport chain. However, under anaerobic conditions, under anaerobic conditions without oxygen, NADH gets converted back to NAD through anaerobic mechanisms. Anaerobic conditions in yeast convert pyruvate to carbon dioxide and ethanol, and this occurs with the help of pyruvate decarboxylase, which is an enzyme that removes carbon dioxide from pyruvate to yield acetaldehyde. The acetaldehyde is then reduced by the enzyme alcohol dehydrogenase, which transfers hydrogen from NADH to the acetaldehyde to yield NAD and ethanol. And this enzyme isn't found in humans. Anaerobic conditions lead to glycolytic products other than pyruvate. So these different products are necessary because the NADH molecule must be reoxidized so that it can function in the next run of glycolysis with the newly introduced glucose. If oxygen is not present to help oxidize it, other reactions such as those of homolactic and alcoholic fermentation must occur. As mentioned before, yeast produces ethanol and carbon dioxide during fermentation. Yeast can ferment different sugars, however, glucose is the most preferred substrate. So in this experiment, we tested for yeast relative fermentation at rates of different sugar. So to do this, we first had 3 beakers. The first beaker was filled with glucose, the second beaker was filled with starch, and the third beaker had starch mixed with amylase. We also had 3 pieces of cake yeast, each weighing 5 grams each. Half a gram each, and 3 swan tubes. along with a 37-degree water bath. So in this experiment, our basic goal was to measure the amount of carbon dioxide bubbles generated to give us an indirect measurement of carbon dioxide produced from fermentation. We hypothesized that the mixtures containing the glucose and the starch only would generate carbon dioxide, indicating that fermentation took place, and that the mixture containing starch with the amylase would not give off carbon dioxide. So for our procedure, we started off by having the 3 beakers, and we took one of the beakers that contained starch and we added 15 milliliters of amylase to it. And then we let that settle for about 15 minutes. After that, we took the 5 grams of the half gram of cake yeast, and we put half a gram of cake yeast into each beaker. We stirred it until it was a homogeneous mixture, and then we added each of the solutions to a separate swan tube. All 3 tubes were put in a 37-degree water bath for about an hour, and every 10 minutes we would periodically check and measure each swan tube to gather the rate of fermentation. So these were our results. After 10 minutes, the starch in the amylase had 0 centimeters of carbon dioxide bubbles. The starch also had 0. Glucose had 0.4 centimeters. After 20 minutes, starch amylase had 0 centimeters, starch had 0 centimeters, glucose had 2.3 centimeters. After a half an hour, starch amylase still had 0, starch had 0, and glucose had 4.6. After 40 minutes, starch and amylase had 0, starch had 0, and glucose had 7.1. After 50 minutes, starch and amylase had 0, starch had 0, and glucose had 9.6. After an hour, starch had 0, starch with amylase had 0, and glucose had 13. So as we can see here, the amounts went up really fast for the swan tube containing the glucose, indicating that fermentation occurred in that beaker. And however, the other 2 tubes containing the starch and the starch with amylase did not have any results. So we know that yeast, like most organisms, can break down monosaccharides. Examples of these are glucose and fructose, and disaccharides like sucrose and maltose, because they're water soluble. This is shown in our results because the swan tube filled with glucose indicated the release of carbon dioxide that shows that fermentation took place. However, we see that the tube containing starch and the tube containing starch with the amylase did not undergo fermentation. So the reason for this is that starch is a disaccharide, and yeast, according to our experiment, yeast demonstrated its inability to break down starch. Because disaccharides are large, complex molecules, the starch cannot be absorbed for nutrients because the sugars they contain are linked together and cannot be dissolved as easily due to their increased size. So, furthermore, this proves our hypothesis to be partially wrong. We did say that the beaker containing the glucose would undergo fermentation, and right here we can see that it did. And we also said that the beaker containing the starch would undergo fermentation, which was tube 3 right here, but it didn't. And this, we can see, although it looks like it kind of did, those are just bubbles, air bubbles of the starch. So, I mean, the liquid is still there. It's just the the yeast settling in. And so basically, it was found that the beaker containing the glucose underwent the fermentation and starch didn't. And that's basically it. Thank you for watching.